Hi, my name is Billy the Neuron, and I'm going to draw my life. To start off, I have a cell body that contains all my organelles, including my nucleus. Then I have an axon that attaches to my cell body. That is what transmits the signals I want to send to other neurons. Oh, and don't forget the axon hillock that connects my axon to my cell body and generates my signals. On the very end of my axon, I have synaptic terminals that allow for the formation of a synapse, which is the region between me and my other neuron friends. That is where chemical messengers called neurotransmitters pass on my information. I also have myelin sheets that cover my axon because I am part of a vertebrate. These myelin sheets allow me to transfer my information quickly, even though I have a thin axon. Oh, and never forget my dendrites, which receive information from other neurons.
Now let's move to more things about me, the things that go on inside of me and around me. First, we will begin with depolarization. Now, depolarization is the reduction in the magnitude of the membrane potential. Hyperpolarization is the increase in the membrane potential. Action potential is the massive change in membrane voltage. Membrane potential is the voltage across the plasma membrane. The resting potential is the minimum potential of resting neurons. The threshold is the minimum membrane voltage required to reach the action potentials. Repolarization is the change in the membrane voltage to turn back to negative values. The refractory period is the downtime following an action potential when a second cannot be initiated. And the undershoot is the final phase of an action potential. Now that we had that out of the way, let's move on to more about me, Billy the Neuron. Now, this is a graph of my action potential. On the y-axis, we have membrane potential, and on the x-axis, we have time.
We start off with the resting potential, which is approximately 70 millivolts. This part of the graph that's increasing represents the depolarization of the membrane. When my membrane potential reaches a threshold value, my action potential occurs. The part of the graph that's decreasing represents repolarization. When hyperpolarization occurs, I enter my refractory period, or the undershoot. Then my membrane goes back to resting potential. Next, let's take a closer look at my membrane proteins to see exactly how the different ions move in and out. The ion channels allow the diffusion of potassium ions and sodium ions, which generates the concentration gradient, which creates a resting potential. There are always more open potassium channels than sodium channels. The sodium-potassium pump uses ATP to actively transport sodium out of the cell and potassium into the cell.
Alright, so moving on to what happens in the gaps between the myelin sheets in my axon. This area is called nodes of Ranvier, where action potential is generated and travels to the next node, where the membrane is depolarized and is produced again. In simpler terms, it seems as if action potential jumps along the axon from node to node. This is called saltatory conduction. Once information is passed through my axon, a chemical synapse occurs. Here, the presynaptic cell meets with the postsynaptic cell. The vesicles that carry the neurotransmitters release them into the synaptic cleft, where it binds to the ligand-gated ion channels. This opens the channels and allows both sodium and potassium to diffuse through them.
And that is the story of my life. Thanks for watching.